Okay, let's unpack this. We're diving into, well, one of the biggest questions in developmental biology. How does a single stem cell know what it wants to be when it grows up?
Exactly. And we're focusing on hematopoiesis making blood. For years, the story was, pretty neat and tidy.
It was a clean family tree, right? You start with a stem cell, it becomes a CMP, then a GMP, and so on,
Hmm.
all defined by a few simple markers on the cell's surface.
That was the classical model. It was elegant, it made sense. But then single-cell genomics just blew that picture wide open.
Ah, right. It suggested things weren't so discrete, that maybe it was all just one big blurry continuum of cells slowly changing.
And you can see why that idea took hold. It explains some of the flexibility you see in the system. But the paper we're looking at today, it really pushes back hard. It brings compelling evidence that, no, it actually is a series of discrete states.
But they're just states we couldn't see before.
We couldn't even dream of seeing them. And the tool that lets them do it is a new framework called ChromLinker. Our mission today is to figure out how ChromLinker settles this debate by finding these hidden, super important cell states they've named MultiLin.
And to do that, they had to look at 3 different layers of information at once.
That's the key. They blended the RNA, which is the cell's active gene list, the surface proteins, which are like the physical badges a cell wears, and maybe most importantly, the chromatin.
The chromatin being the blueprint, the master plan of which genes are even available to be turned on.
Precisely. It's what the cell is capable of doing. And if you can define a cell by all 3, what it's doing, what it's wearing, and what it's capable of, then you can prove it's a stable, unique stage.
Not just some random point on a blurry line.
The first big problem wasn't even about the biology, it was about the technology. The tools were sort of working against each other.
Right. You had flow cytometry, which is great for sorting millions of live cells based on their surface badges, but you're limited to just a handful of those badges.
And on the other hand, you have these amazing single-cell sequencing methods that tell you everything happening inside the cell.
But you have to destroy the cell to read the blueprint.
Exactly. You get this perfect picture of a ghost. You know it was there, but you can't grab it. You can't watch it grow up. It's gone.
So they needed to build a bridge between those 2 worlds. Let's start with the tools they used to build the map itself. First up, CITE-seq.
CITE-seq is the first big piece. It's multimodal, so it measures 2 things from the same cell, the RNA and hundreds of surface proteins using these things called ADTs. But the crucial part here is the incredible amount of work they did just optimizing the tool.
What do you mean? They couldn't just use an off-the-shelf kit?
Not if they wanted the data to be bulletproof. They talk about this painstaking process of molecular titration. They started with 195 antibodies and manually optimized and refined them down to a core panel of 103.
Why go through all that trouble?
Because if your goal is to overturn a major theory like the continuum model, your measurements have to be flawless. And that effort paid off.
No.
Using this hyper-optimized data, they identified, get this, 87 stable discrete cell states.
Wow. Okay, 87. That right there is a huge blow to the simple continuum idea.
It really is. So that's RNA and protein. But they still needed that 3rd layer, the chromatin blueprint.
And that's where TEA-seq comes in.
That's where TEA-seq comes in. It's a trimodal assay, which is just phenomenal. From one single cell, it measures RNA, surface protein, and the chromatin accessibility, the ATAC-seq data. So now you have the complete picture.
You know what the cell is doing, what it's wearing, and what it's ready to do next.
You got it.
Okay, so they've built this incredibly detailed genomic atlas, but the ghost problem remains. How do you find these 87 states in a living, breathing population of cells you can actually sort? That must be where InfinityFlow comes into play.
InfinityFlow is the brute force, high-throughput flow cytometry. It measures 140 surface markers on millions and millions of cells. So you have a physical atlas based on protein, and now you have the big central challenge.
The translation problem.
The translation problem. You have your genomic clusters from CIE-seq, which are defined by, genomic language A, and then you have your physical protein signals from InfinityFlow, which is protein language B. The units don't match. They aren't even in the same ballpark.
So how do you tell the sorter, hey, find me the cells that match the genomic profile of cluster 42?
This is where ChromLinker becomes the hero. It's a really clever 2-step normalization strategy they call KDE mapping.
Okay, KDE mapping. Break that down for us.
Think of it like trying to compare temperatures in Celsius and Fahrenheit without a conversion formula. The raw numbers are meaningless. But if you talk in percentiles, you can find a common language.
So you could say the hottest 10% of days in both systems, and that would match up.
That's exactly it. It converts the signal intensities from both platforms into percentile ranks. So the top 5% of cells for a certain marker in CITE-seq can be directly mapped to the top 5% of cells for that same marker in InfinityFlow. It creates a universal exchange rate.
And that lets them transfer the super high-resolution genomic labels onto the physical sortable protein map.
And suddenly, you can physically isolate cells based on their internal genetic state. The ghost now has a body.
All right, let's take a quick checkpoint here. So the whole continuum idea is looking shaky because this new atlas shows 87 stable, discrete cell states.
Right.
And the technical breakthrough, ChromLinker, uses this clever KDE mapping to bridge the gap between destructive genomic data and practical physical cell sorting with InfinityFlow.
Which finally let them hunt down and capture these rare, previously invisible multi-Lin states.
So now the big question. These MultiLin cells were hiding inside the old, messy CMP and GMP gates. Now that we can isolate them, what's actually going on inside? What's the genetic logic that separates them?
Well, the first clue to finding their home came from looking at the surface markers again. The InfinityFlow data showed one glaringly obvious signal across all these new multi-Lin populations.
Which was?
They were all universally CD27 high. That became the key. The sorting gate, Linsky1 CD117 plus CD27 plus, was the doorway to this hidden developmental crossroads.
So CD27 is like the signpost for this entire transient stage.
It seems to be. But isolating them is just step one. To really understand the discrete state model, you have to decode the software, the gene regulatory network, or GRN, that's making the decisions.
How do they read the code?
They took that TEA-seq chromatin data and fed it into a deep learning model called ChromBNet. This thing is incredible. It doesn't just see that a region of DNA is open, it looks at the pattern of openness and can predict, down to the single base pair, which DNA sequences are acting as the master switches.
So it's like a forensic tool that finds the exact fingerprints of the master regulators, the transcription factors.
That's a great way to put it. And when they ran this analysis, the result was crystal clear. They saw a fundamental bifurcation, a two-way split in the road.
Driven by different sets of these master regulators.
Exactly. On one path, in the cluster they called ML2, they saw the activity of Q.1 and IRF8 ramping up. These are the classic conductors of the myeloid orchestra. They tell cells to become macrophages, neutrophils, dendritic cells.
And the other path?
That was in cluster ML3. Here they saw the nascent activity of Geta1 and Geta2. These are the undisputed kings of the erythroid and megakaryocytic lineages, the ones that make red blood cells and platelets.
So this is the first time we can actually see these two opposing forces at work in two distinct, physically isolatable groups of cells.
Yes, and this leads to the real moment, the discovery that connects this hidden internal software to something you can see on the outside.
The badges.
The badges. They found that a protein called CD371 is expressed almost exclusively on the ML2 cells, the myeloid primed ones. And its expression is directly tied to that IRF8 and Q.1 activity.
So if a cell is wearing the CD371 badge, you know the myeloid machinery is already running inside.
Precisely. And conversely, a different protein, CD55, is expressed on the ML3 cells, the erythroid primed ones. Its expression is linked to the gain of factor activity.
That's amazing. So you don't need a multi-million-dollar sequencing experiment anymore. You can just look for these two simple surface markers, CD371 and CD55, to know which path a cell is already starting down.
It gives you a direct readout of the internal commitment state. It's a massive leap forward.
Okay, the computational story is incredibly compelling, but this is biology. You have to prove it in a dish. How did they validate that ML2 and ML3 were actually restricted in what they could become?
They hit it with two gold standard functional tests. The first was lineage tracing with a system called cell tag.
That's where you barcode individual cells and then track where their descendants end up, right?
Yeah.
Like a family tree.
It's exactly like a family tree. They put these viral barcodes into the progenitor cells and watched them grow. And the results were unambiguous. They saw a very strong link, a high clonal coupling score between the ML3 cells and the mature megakaryocyte and erythroid cells.
And I'm guessing a similar link for ML2.
A very strong link between ML2 and the monocyte and neutrophil progenitors. It was physical proof that these are truly divergent paths starting from these distinct multi-Lin states.
Okay, that's one test down. What was the second?
The second was the ultimate proof, a functional potential assay. This is where they really put them to the test. They used InfinityFlow to sort single cells based only on those new CD55 and CD371 badges.
So they grabbed the CD55 positives, the CD371 positives, and the ones that were negative for both.
Yep. The double negatives would be the earlier ML1 state. They put each single cell into its own well and just let them grow for 5 days to see what they'd become.
And the results.
Were stunningly clear. The double negative cells, the ML1s, were genuinely multipotent. They could make everything. Erythroid cells, eosinophils, neutrophils, you name it.
As you'd expect from an early progenitor.
But the CD55 positive cells, the ones skewed toward ML3, they were almost completely restricted to the erythroid fate. And the CD371 positive cells, the ML2 skewed ones, they were locked into making myeloid shades. It's the smoking gun. The external badges perfectly predict the internal functional fate.
And this whole system isn't just a static map, is it? It has to respond to real-world needs, which brings us to the infection model.
This is where it all comes together. They used a mouse model infected with a parasite, Nipistronilus brasiliensis. This type of infection requires a massive, rapid production of basophils and eosinophils. The question was, which of these progenitor states gets the call to action?
Did the whole system just ramp up, or did the response come from one of those specific pathways?
It was incredibly specific. During the infection, they saw a huge increase in the CD55 positive ML3 gate. And at the same time, the CD371 positive ML2 gate actually went down.
Wow, so the body specifically taps into that ML3 reservoir to fight this kind of infection. It's the go-to factory for making basophils under stress.
It confirms that ML3 isn't just a computationally defined cluster, it's a physiologically critical precursor state that the body actively regulates.
Let's do a quick final recap of the core findings then. This is a lot to take in.
For sure. So, 1. The ChromLinker framework bridges the gap between genomics and physical cell sorting, basically settling the debate in favor of discrete, stable states.
2. These multilin states, ML2 and ML3, are real, isolatable, hierarchical stages.
3. Their different fates are determined by a battle between master transcription factors GATA on one side, IRF, and SPI on the other.
And 4, and this is huge for practical use, CD55 and CD371 are reliable surface reporters for these hidden internal states.
And finally, these states are not static. They dynamically shift to respond to real-world stress like an infection.
So, stepping back, what does this all really mean? It feels like we've traded a simple but wrong map for a much more complex one that's actually correct.
That's a good way to put it. The source argues that development isn't a vague continuum. It's a highly organized, hierarchical process made of discrete states, each defined by the complete integration of its internal software and external hardware.
And the ChromLinker framework itself, that seems like the biggest takeaway. It's a general-purpose solution, isn't it?
Absolutely. This whole idea of using computational mapping to link multi-omic data with physical sorting, you can apply that anywhere. Cancer, neuroscience, immunology. It's a roadmap for finding and isolating critical rare cell states in any system.
What's really striking is they mention finding these same kinds of shifts in the multilin populations across 13 other published stage vets, from aging to different diseases.
Which brings us to a really provocative thought for you to chew on. If ML3, the CD55 positive group, is biased toward making things like basophils and red blood cells implicated in allergies and anemias, and ML2, the CD371 group, is biased toward myeloid cells implicated in inflammation and some leukemias.
Then could you imagine a future where monitoring the simple CD55 versus CD371 balance in a person's blood could be an early warning system?
Exactly. Could tracking the balance at that key bifurcation point give you a diagnostic insight into a developing disease long before any major symptoms appear? It could turn a fuzzy clinical picture into a sharp, actionable data point.
A fascinating thought. For anyone who wants to dive even deeper and explore this incredible atlas themselves, all the data is publicly available in the Marrow Atlas resource.
It's definitely worth a look. The sheer scale of the data is something to behold.